it's not just you in the hall, but it's more than 300 other experts and people interested in the field in the stream, to welcome you to this first collaborative meeting of Christian Brecker and Professor Lacroix. So thank you very much for your initiative and for pushing this field, which is so important. We all know that there is plenty of microorganisms in the gut, but it's not just for the gut specialists. We have here quite a bit of gut specialists in the audience, but we have also brain specialists and other specialists. So the microorganisms in the gut play a role for almost everything in the human body, and it's quite dynamic over time. So it's wonderful to have excellent experts talking about how this changes over the age of infants and children. And I think we will have wonderful 3 hours with excellent discussions. So thank you very much, Christian. Thank you very much, Professor Lacroix. The floor is yours.
Thank you very much, Michael, for the nice welcome. And I would also like to thank you all here in the audience, and also the ones that are online, who join us online for this meeting. You may ask yourself, what is the background of this meeting? Well, we actually have a very long-term collaboration. It's actually 14 years between the Division of Gastroenterology and Nutrition and the Nutrition Research Unit of the University Children's Hospital in Zurich, with the Laboratory of Food Biotechnology of the Institute of Food Science and Nutrition of the ETH. And we wanted to demonstrate the results of that collaboration of the last 14 years. During this time, we had 5 PhD students. Alina Zieler, she finished in 2010. Then Ted Joost, Van Pham, Vanessa Rocha, and currently Nise Otaru. They all did very well. They were very successful. They had a lot of publications in highly ranked scientific journals. And Vanessa Rocha, who is here in the audience, she actually, in addition, won the ETH Pioneer Fellowship in 2019. She is currently developing a first microbial solution for infant colic based on a clear mode of action. The product that she will launch is called Bactokind, and Christophe Lacroix will talk about it a little bit more later on. Before I now start with the first presentation, I would like to thank my current team at the Children's Hospital in Zurich. It's the current team that has been involved in these studies. It's the PhD student Nise Otaru and the 2 study nurses, Rebecca Koller and Vanessa Rüeger, who are very important for this sort of research because they are the interface between the laboratory and the probants, the mothers and the infants, and they did really a great job over very many years. So thank you very much. I would also like to thank Vanessa, in addition, for helping in organizing this meeting. Without her, we would not be here. She made everything. The program was made by Christoph and myself, but the rest actually was done by Vanessa, including the very nice website. So thank you very much. And then, last but not least, I would like to thank Michael Grotzer, again, the medical director of the University Children's Hospital, for supporting us for very many years and for the time being. We are very grateful for that, and I would like to thank you again. Then we did not want just to demonstrate what we have done in the last 14 years together with ETH, but we also wanted to put it in a greater context. And that's why we invited 3 of the most prestigious and most successful scientists in this field from all over from Europe: Catherine Stanton, Maria Carmen Collado, and Paolo Leonetti. And the first presentation will be held by Catherine Stanton. She is a Senior Principal Research Officer at TIGASH and APC Microbiome Institute in Ireland, and a research professor at the University College in Cork in Ireland. Her scientific focuses are probiotics in human nutrition and health, functional foods and human microbiome, influence on dietary and environmental factors on infant gut microbiota. She has published a tremendous number of publications in excellent journals. Her Hirsch index is incredible, 120, and she has been cited nearly 60,000 times until now. So I would like to ask you to come to the floor. Thank you very much. Yes, you need to use this. It's like that, yeah.
Like this?
Yes.
So I'll just speak away then.
I think it's working.
Yeah. Just like this. And I have 45 minutes. Thank you. So thank you very much, and thank you very much to the organisers for inviting me here today to speak with you, microbiome from infancy to old age. So I'm going to take a look at the factors influencing gut microbiota development in the early stage of life through to old age, and what factors influence it and the potential to modify it as a result of those exposures. I suppose over the last number of years in particular, it's been well recognized that the gut microbiome is integrally integrated with human health and development. And we are gaining a continued increasing appreciation of the role of the gut microbiome in various aspects of human health. So just to start off, let's talk a bit about what the gut microbiome is and how it relates to health and development. So I suppose the gut microbiota is, well, in fact, the human gut is one of the most densely populated ecosystems on the planet, and contains not only bacteria, but various other forms of life, including fungi, archaea, etc. But in terms of the composition of the human gut microbiota, we each of us harbour in our intestines up to 10 to the 14 bacteria per gram of gut bacterial contents, consisting of at least, but probably a lot more than 1,000 different bacterial species. These are primarily consisting of Firmicutes and Bacteroides, which dominate the gut microbiota. And indeed, however, there is a vast inter-individual variation in the composition of the gut microbiota of each different human being, such that no two people are actually alike. The composition of the gut microbiota is influenced by a vast array of factors that we are exposed to throughout life. In particular, dietary factors, which of course we continue to be exposed to on a daily basis, environmental factors, and age, which is indeed the topic, I suppose, of my presentation today. And it's generally accepted that a diverse microbiota composition and function is important for ongoing health of the human being. The gut microbiota has a number of functions that it performs for the host on an ongoing basis. And indeed, gut microbiota is integrally related to host health and has an integral association with metabolism, intestinal function, indeed bone homeostasis, and indeed brain function and behavior. So in terms of functional aspects of the gut microbiome, it's integrally involved in the development of the immune system, and that's particularly appreciated in the early phase of life when the infant is born and is exposed to his first microbes. This is strongly influencing the optimum development of the immune system in the early period following birth. In terms of functionality, the gut microbiota has an important role to play in digestion of complex carbohydrates, and indeed many of these complex carbohydrates are indigestible to the human itself, and we do not have the enzymes to possess, we do not possess the enzymes to digest some of the carbohydrates which we eat. However, the human gut microbiota do possess those enzymes and conducts digestion of these complex carbohydrates on our behalf and for our benefit, leading to the production of more energy for the host, and also influencing the production of various bioactive molecules, including short-chain fatty acids. So the gut microbiota produces a range of helpful benefits for us, including the production of a variety of beneficial microbial metabolites, such as vitamins. For example, B vitamins and vitamin K are produced by the gut microbes, and of course are important nutrients for human nutrition. And also short-chain fatty acids, which are signaling molecules, as well as energy for colonocytes, are primarily and uniquely produced by gut microbes upon their digestion of complex consumed polysaccharides by the human host. The gut microbiome also exerts protective effects in the intestinal tract by protecting against the invasion of potentially pathogens, and also contributes to the maintenance of gut barrier function. So a healthy gut microbiome is obviously very beneficial for the human host, but on the other hand, in terms of the negative consequences, a perturbed gut microbiome, which can be perturbed by a number of factors that I will talk about today, are seen in various disease states and are caused by various environmental exposures. For example, a perturbed gut microbiota associated with inflammatory bowel disease, irritable bowel syndrome, obesity, diabetes, autism, and depression have demonstrated that the perturbation of the microbiota can lead to the onset of diseases in the human host. So in terms of numbers, looking at the gut microbiota in terms of numbers, shown on this slide are some of the amazing numbers that are associated with the human gut microbiome. For example, we contain 100 trillion symbiotic microbes that live in and on each person and make up the human microbiota. That's 100 trillion different living bacteria in and on the human body. Indeed, the human body has more microbes than there are stars in the Milky Way. And 95% of the bacteria on the human host are actually residing in the gastrointestinal tract, demonstrating the significant involvement and ecosystem that lives in the human gut and contributes to host health. There are 150 times the number of genes in our microbiome than there are in our human genome, clearly demonstrating the intense metabolic capability of the human gut ecosystem that lives in the human gut. So they don't consist of just bacteria. There are 5 times the number of viruses as opposed to bacteria in the human gut, including the phagome and virome. But I guess I won't be talking too much about that today. I'll focus more on the microbiome, the gut bacteria living in the gut. So the microbiome is malleable and manipulable, more so than than the human genome, and as a result can be changed, unlike the host genome. As I've said before, it's integrally linked to human health, and so its manipulation in a positive sense can potentially have be a new therapeutic intervention for enhancing the health of the host. And factors such as age, diet, host, and antibiotics shape the microbiome, but not precisely. And so a number of research approaches these days are looking at how we can manipulate the microbiome more precisely to positively impact on human health. So let's start at the beginning, and in early life, the first 1,000 days from conception up until about the age of 2 years are known to be an intensely important period in early development. And indeed, during this period of time, but in particularly around the time of birth, intestinal microbiology becomes important, and the early life establishment of the gut microbiome following birth establishes a symbiosis between the gut microbes and the human host in the early period and leading on into later life. So in this early period, the early microbiota that seed the newly born infant impacts health in a very important way, in particular programming the immune system and the development of the immune system in early life in a positive way. An altered microbiome in early life as a result of things such as antibiotics, mode of delivery, etc., early exposures can negatively impact the pioneering microbes and their development and acquisition, which can lead to negative consequences for the infant and for the developing child, including allergy and other disorders such as diabetes, etc. So this early period of microbiome seeding is important and is a subject of intense investigation, as you will hear some of the results of some of the studies today. So I suppose we'll go on to the next slide. Not only do the early microbes impact on the immune system, but also brain does not actually develop normally without gut bacteria. And the gut-brain axis is influenced and regulated by the microbiota and driven by microbiota gut-brain axis interactions. Animal studies, in particular studies involving animals without a gut microbiota, i.e. germ-free animals, have demonstrated this in a number of studies, demonstrating the integral role of the gut microbiome in various aspects of neurogenesis and the development of the neuronal system, with impacting on myelination, availability of neurotransmitters, neurogenesis, and dendritic growth. So let's look at some of the perinatal factors that affect the gut microbiota in the early period of life. This slide summarizes the dynamics, really, of microbial exposures in the period before birth, i.e. during pregnancy, and in the early period following birth. So what you can see is that in terms of the pregnancy, there are alterations associated with pregnancy. For example, the vaginal microbiome and the gut microbiome are reduced in diversity during the pregnant phase, and then subsequently expand again upon delivery of the infant. Breast milk microbiome, which we'll hear more about later, is an important seeding factor as well for the infant, for infant nutrition. And the composition of breast milk changes throughout lactation, being more diverse in the early period and subsequently reducing in diversity as lactation progresses over the months following birth. There are a number of dynamic changes that occur to microbes in the gut and in the vagina. For example, in terms of during the pregnancy, actinobacteria, proteobacteria are increased, but in the gut bacteria, whereas some of these alterations change again once the baby is born. And so there's a kind of a dynamic arrangement or ongoing situation between pregnancy and post-pregnancy. And the gut bacteria in particular, but also the vaginal microbiome, are particularly important in terms of mother-to-infant transmission, which contribute not only to the microbes received at birth, but also the early seeding microbiome from birth onwards. So you can see on this slide the three circles with the infant following birth, i.e. the birth period, then at 1 month and at 4 months. And what this is demonstrating is the enduring effect of early life exposures on the early microbiome development in the months following birth. So what's clearly evident is the the immediate effect of maternal gut, shown in blue on the circle, which is both immediate in the sense that it impacts the composition of the infant gut at birth, but it's also enduring in that it continues to have an influence at 1 month and at 4 months, and a significant influence at that. In addition, there are other factors that have shorter and maybe less enduring influences. For example, the maternal vagina, the impact of that or its contribution to the seeding of the infant gut, shown in green, is important at the time of birth in the vaginally born baby, but is less so as the months progress after birth. Maternal skin is another component there, shown in red, where it has a very small influence around the time of birth and less so as time progresses. So this just summarizes the maternal factors, in particular the seeding of the early life gut microbiome by the human maternal gut at the time of birth, and its enduring effects on the ongoing development of the infant gut microbiome. And all of these factors are in the developing early life gut microbiome are influenced by factors such as the mode of delivery, which we'll talk about in a moment, antibiotic exposure, gestational age at birth, the diet obviously, and host genetics. And these are the main factors known to have significant influences on composition and functional activity of the gut microbiome following birth. So looking at the impact of birth mode on gut microbes, I've just shown you here a study by Domingos Bello et al., which was published a few years ago now, demonstrating the importance of delivery mode in that very early period following birth. And she and colleagues demonstrated that delivery mode shapes the acquisition and structure of the initial microbiota across multiple body sites in newborns. So the slide shows really that the mother's microbiome, in terms of her skin microbiota, significantly influences the C-section-born infant gut microbiota immediately after birth. And indeed, C-section-born infants were found to be dominated by skin-associated bacteria such as Staphylococcus, Corynebacterium, and Propionibacterium, which are microbes associated with skin. And these were the early colonizers in the babies born by C-section. On the other hand, bacteria such as Escherichia shigella and Bacteroides were underrepresented in the infants born by C-section compared with vaginally born infants. And vaginally born infants' microbiome was more akin to that of the mother's vagina immediately after birth, which have a predominance of Lactobacillus, Prevotella, and Schniatia species. Infants born by C-section were also found to have a lower bacterial richness and a lower bacterial diversity compared to vaginally born babies after the birth process. So anyway, this demonstrates that birth mode, whether a baby is born by natural delivery or taken up by surgery in terms of C-section, has a significant immediate impact on the early seeding microbes that are found in the human gastrointestinal tract in the hours and days after birth. And then taking a look forward at the longer-term impact of that birth mode up to 1 year of age. This is a study we conducted a couple of years ago where we looked at shotgun metagenomic data of published studies and comparing and looking at the shotgun metagenomics of mother-infant pairs to look at the impact of birth mode on the developing microbiome in the infant up to 1 year of age. So you can see clearly on the left-hand side of your screen the vaginal microbiota, and the box to the right of that is the C-section microbiota in comparison. And I suppose the first thing that's clearly obvious is that look at all the missing microbes that are missing in the C-section born infant gut over the first 12 months of life compared with the vaginally born microbiome. So C-section drastically reduces transmission of microbial taxa from mothers to infants compared to vaginal delivery. Indeed, we found only 2 species that were consistently transferred in C-section delivered babies, as opposed to 22 species in the first 12 months of life in the vaginally born babies. So therefore, again, this demonstrate at a metagenomic level the impact of birth mode in terms of microbiome acquisition and also the functional consequences that this has for the developing infant over the first year of life. This meta-analysis of mother-infant fecal metagenomes we also studied it in terms of the comparing the mother microbiome with the infant microbiome. You can clearly see on your left side of your screen there an overview of the composition of at the genetic level of the microbiota in the pregnant woman and at delivery in the first 3 columns, whereas after that you can see the infant microbiome. And you can clearly see differences, quite significant differences in the microbiota of the mother versus the infant, where the mother microbiome is dominated by Bacteroides, Eubacterium, Alistipes, Ruminococcus, Faecalibacterium, Prevotella, Roseburia, microbes like that. As a result, as she's obviously an adult, and so that's a description in a way of the adult microbiome. Whereas on the other hand, the infant microbiome is shown from at birth, week 1, month 1, month 3, 6, and 12. And you can see that there's an evolution of the microbiota during this period, where the bacteria shown in blue are more prominent in the infant gut microbiome compared with the mother microbiome, and are enriched in Bifidobacteria, Escherichia, Lactobacilli, Clostridium, and Parabacteroides compared with the maternal microbiome. You will notice as well that by the age of 12 months, the infant microbiome is getting closer in terms of overall similarity to the maternal microbiome, but it still hasn't reached maturity by 1 year of age. In terms of metabolism, there were also differences noted between mothers and infants, and also differences noted as a result of the birth mode of the infant, whether it be naturally born or C-section born in the early period of life, with differences in metabolic functions between mothers and infants clearly observed as a result of age of the infant. So infants' microbiome were more enriched in fermentation of human milk oligosaccharides and such complex carbohydrates in the early microbiome compared with the maternal microbiome. Here I'm just demonstrating a study, showing you some data from a study that we call the InfanMet study that we published a few years ago, looking at the evolution of gut microbiota composition from birth to 24 weeks as a result of birth mode, but also preterm gestational age at birth. So we compared the microbiome development of the full-term vaginally delivered babies versus full-term C-section delivered versus preterm babies. And I suppose just to note, again, in terms of a comparison of the full-term vaginally delivered infants versus the full-term C-section delivered infants, I suppose what's noteworthy is the reduced blue bar component, which is the actinobacteria or bifidobacteria that are really deprived in that early period following birth as a result of C-section birth mode, but they're already naturally there in ample components, ample relative abundance, even at week 1 of life as a result of vaginal delivery. Furthermore, the bacteroidetes component in the C-section infants never really caught up even by week 24 compared with the vaginally born infants. And then comparing those with full-term infants with preterm microbiome, you can again see further differences as a result of preterm birth. And I suppose in particular to note, especially immediately or soon after preterm birth, the high relative abundance of proteobacteria, which are potentially negative for human health, associated with the early gut of the preterm infants. Looking then at, continuing to look at the InfanMet data, you can see that irrespective of birth mode and irrespective of whether the infant was born preterm or full-term, microbiota diversity continued to increase over the first 24 months of life, such that there were similar levels of microbial diversity observed by the first 6 months of life. Going back to the metagenomic analysis, we looked at comparing the diversity, or at least continuing to look at diversity up to 1 year of age, and also, first of all, though, just to note on your left-hand side, comparing the microbiome diversity, microbial diversity of the mothers and infants. You can clearly see that the infant microbiome is well reduced in microbial diversity compared to the maternal microbiome, and that's after birth. And on the right-hand side, you can see the increasing microbial diversity, which by 1 year is beginning to catch up, if you like, with maternal microbiome diversity. So microbiome diversity continues to increase in the first years of life. And indeed, this data set just looks at I suppose the dynamics of what's happening in the infant gut over the first 24 weeks of life. Shown in the box in blue. Let me see now, can I? No, I won't do that. I'll leave it. Okay, so the week 1, the genera found in the infant gut at week 1 are boxed in the blue box. And week 24 microbiome is in the red box. So what I'm trying to demonstrate is that week 1 microbiome is enriched in staphylococci, streptococci, propionibacteria, as I mentioned before, corynebacteria, which are in abundance at week 1 of life, but when you go back over to the red box, they're missing. Gone by 24, whereas on the other hand, at week 24, the genera that are present at week 24 were not there at week 1, and include such genera as Blautia, Roseburia, Prevotella, Ruminococcus, which are indicative of more adult-like microbiome. So what this data set, I suppose, demonstrates, if anything, is just the evolution or the dynamics of the microbiome in early life, but also the beginning of maturation, even by the age of 24 weeks. Moving it forward to the first 4 years of life, we continue to look at this data set and found that the microbiome continued to evolve over the first 4 years of life, and indeed So genera, on this slide, genera in the bottom left corner of the map, including Escherichia, Shigella, and Enterobacter, were present at greater abundance at year 1, whereas genera on the bottom right-hand corner, including Christiansonella and Ruminococcaceae, were present at greater abundance at years 2 and 4, whereas Bifidobacterium, which are in high relative abundance in the early period of life, as I showed you up to 24 weeks, continued to disappear or reduce in relative abundance by year 4. So again, demonstrating that even by year 4, things are still changing in the early gut microbiome. It's still continuing to evolve and mature as time progresses following birth. And this shows that the microbiota diversity continues to increase up to 4 years, but also there are differences in the microbiome of infants at year 1, 2, and 4. So each dot represents the overall microbiome composition of an individual. And so what you can, I suppose, clearly see, take from it, is that there is a clear separation of the 1-year and 4-year microbiome, whereas the 2-year microbiome, as expected, would be in between the 1 and 4 years of age. And on the right-hand side there is just a demonstration that the continued complexity and richness of the microbiome continues to increase from year 1 in red to year 2 in blue to year 4 in orange. So there's, again, a continuous complexity developing in the microbiome as the infants progress to 4 years. Going back to the preterm infants, we compared them in terms of their microbiome with the full-term infants at 4 years of age, and shown on the left-hand side of your slide is that there's still a separation, actually, of the microbiome composition of the preterm, of the infants that were born preterm. They're now 4-year-old children, but they still separate in terms of microbiome composition 4 years later. So this demonstrates that the microbiome actually has a memory of premature birth, even 4 years on. And so what the implications are that for nutrition in terms of the functionality of the microbiome and how it impacts on the developing host need to be considered. Recently, we have published a description of fecal metagenome analysis of children under 3 years of age. So there was an age-specific reference genome of the human gut microbiome can provide higher resolution for genomic analysis, including taxonomic classification, strain-level genomics, investigation, and functional characterization. So this presents the early life gut genome catalogue of over 32,000 metagenome assembled genomes that are publicly available, which we stratified based on age up to 3 years, gender, delivery mode, gestation, and the feeding regime. And we conducted a number of bioinformatic analysis to interrogate various aspects of early life exposures in terms of microbiome genomes. So I suppose just a couple of things from this publication. The key Bifidobacteria species included Bifidobacterium longum, which was most prevalent and exerted a number of different functionalities to the microbiome. There were a number of MAGs that were both in high abundance and also in terms of strain heterogeneity, were more simple in the early period following birth and then became more complex later. We found that Bifidobacterium in particular, Bifidobacterium longum subspecies infantis harbored a significant contribution to metabolism of the human milk oligosaccharides found in breast milk, and these were enriched in the early life gut microbiome. And shown on the bottom hand of your left side is the glycobiome. In other words, the functional repertoire of these Bifidobacterium to metabolize these human milk oligosaccharides, and you can clearly see the rich repertoire of genes involved in the genome that are suitable for metabolizing the early life feeding regime of breast milk oligosaccharides. In terms of delivery mode, the study demonstrated the enduring effect of C-section birth mode on the microbiome over the first 3 years of life. And one thing that we noted was the more enrichment of antibiotic resistance genes associated with C-section delivery compared with a vaginal delivery. And this could be due to the fact that in most cases, C-section birth mode is associated with antibiotic exposure at the time of birth, and this can have an enduring effect on the infant microbiome, even up to the first during the first years of life. Another aspect is we compared the gut microbiome between children and adults, and this slide just demonstrates that the genome from adults showed higher diversity than from children for 5 of the 12 species studied, including Bacteroides fragilis, Bacteroides ovatus, Bifidobacterium, Bifidum salivarius, and Streptomophilus. This indicates that the genomes within these species in early life are more conserved, but in later in life, more specific genes are required and added by these organisms to perform more functions later in life as the infant, as the diet gets more complex, you know, as the individual grows up. On the contrary, the enriched species B. longum actually showed higher gene diversity in children than adults, and based on my last slide, again demonstrating the importance of the B. longum in terms of metabolism of the early life or the early diet enriched in such things as human milk oligosaccharides for early life nutrition. So I suppose, keeping an eye on the time, in terms of modulation of the gut microbiome, I've told you about the impact of delivery mode, age in the early period, host and exposure to antibiotics shapes the microbiota, but not precisely. And so there is an interest in therapeutic modulation and how can we positively modulate the early life microbiome using diet and therapeutic interventions. So first of all, just looking at breastfeeding, which I've mentioned already. Obviously breastfeeding is the optimum and gold standard feeding regime for infant nutrition and contains all the right nutrients in the right proportions over the first months of life. But in the InfanMed study, we looked at the impact of breastfeeding of the microbiome of the C-section versus vaginally born infants and found that breastfeeding had a greater effect on the C-section microbiome than it did on the naturally delivered microbiome. There were a number of genera were different in those that were breastfed for longer, demonstrating that it drives breastfeeding drives microbiota maturation more significantly in C-section born infants than vaginally born infants. Just briefly looking at milk microbiome composition, we conducted a study where we analysed the milk microbiota of donors. 231 breast milk samples were analysed throughout lactation from week 1, 4, 8 and 24 weeks. And this study demonstrated that the milk microbiome composition changes throughout lactation and separates based on stage of lactation. So you can see clear differences in microbiome composition of the milk at week 24 of lactation, for example, compared to the colostrum or early milk microbiome. And otherwise, milk microbiome composition was not impacted by such factors as whether the birth was a C-section or vaginal birth by the mother, or whether the baby was a male baby versus a female baby, and some other factors as well. So this demonstrates that microbiome composition, milk microbiome composition changes throughout lactation, and this, of course, feeds the infant during those first weeks and months of life. And so the breast milk is indeed a source of beneficial microbes and associated benefits for infant health, and has an impact on the maturation of the the gut microbiome and impacts on development of the immune system and other aspects of infant development. Okay, I've talked there briefly about the role of infant nutrition and breast milk feeding, but C-section birth clearly is associated with an altered microbiota, leading to missing microbes in the early period of the infant gut. And a proof of concept study was fairly recently published by colleagues in, a group in Finland that demonstrated in a proof of concept study the role that fecal transplantation could play in seeding the C-section born microbiome to rapidly restore normal gut microbial development. Now, in this cartoon there, you can see that obviously the vaginal birth led to the optimum microbiome of the infant, including enriched Bacteroides and Bifidobacteria, whereas the C-section birth had the altered microbiome, as I've already discussed. But in the middle there, the C-section birth, when it was seeded with maternal fecal microbiota transplantation, led to the development of newborns. Fecal microbiota development of newborns is dependent on the mode of delivery, as I've mentioned, and the fecal microbiota transplantation led to enrichment of the early life C-section born microbiome in a manner closer to the vaginally born. It is a proof of concept study. Fecal microbiota transplantation is not something that is going to be widely acceptable in terms of early life seeding, but it just demonstrates that the microbiome of the maternal gut has a potential to seed the microbiome of these infants, and so therefore we need to think about developing safe, described, well-characterized consortia of microbes that could indeed seed that infant C-section gut microbiome to make it more similar to the vaginally born microbiome shortly after birth. Now, in terms of looking at Bifidobacteria, relative numbers, the Bifidobacteria relative abundance decreases with aging, and indeed Bifidobacteria, of course, is well recognized as a healthy population of members of the gut microbiota. They're very enriched, as I discussed, in the early life gut microbiome, especially in the vaginally born babies. They have a big contribution to human health and HMO and other carbohydrates and the production of various bioactive compounds. But they decline with aging, and so therefore developing populations of Bifidobacteria or consortia of Bifidobacteria to restore the aging process could be a way to repopulate the aging gut microbiome. So Bifidobacterium longum is the most dominant species in infants, but is also present in adults and has a large repertoire of metabolic capabilities suitable for infant and adult metabolism. Looking at Bifidobacterium longum, it consists of Bifidobacterium longum subspecies infantis and Bifidobacterium longum subspecies longum. But you can see that the Bifidobacterium longum subspecies infantis is particularly enriched in gene clusters for HMO metabolism, while the subspecies longum is more specialized for plant-derived sugars with a more limited number of fucosal lactose clusters for growth on 2FL and 3FL suitable for early life nutrition. So looking at the B. longum genome, we found that it was highly variable, consisting of 70% a variable genome with only 30% of the core genome, and isolating and characterizing a large group of B. longum strains allowed us to develop a polybiotic representing the pangenome of the B. longum species, comprising a collection of strains which together constitute the pangenome of that species with all the catabolic and anabolic functions of the genome, of the genus. So in terms of a summary of the early life microbiome part, and hopefully I have a bit more time, the neonatal period is a critical early window for mother to baby transmission of gut microbial strains, with early microbial acquisition being a controlled process rather than occurring by chance. And the colonized early microbes, regardless of their origins, show a better fitness and stability in infants. Cesarean delivery and antibiotic exposure at that early period disrupts that vertical transmission of maternal gut microbiota, including Bifidobacterium and Bacteroides species. And this may predispose colonization of opportunistic pathogens arising from the hospital environment, which can persist into the gut, into infancy and beyond. Breastfeeding has a greater effect on gut microbiome of C-section babies versus vaginally delivered babies. And there is a potential for development of live biotherapeutics or polybiotic solutions to replace the missing microbes in C-section born infants. So, moving on then from infancy to older age, studies have shown that the microbiome continues to develop and stabilizes during adulthood, but again then unravels in old age. And a number of research groups have been looking in particular at the microbiome disruption that occurs during the aging process. So, aging is associated with an unraveling, as I said, of the gut microbiome, and healthy aging associated with enhanced longevity, high physical activity, good diet, and less cognitive decline is a healthy aging process, and the unhealthy aging, on the other hand, where an individual experiences more increased inflammation, frailty, cognitive decline, increased cognitive disorders, and other negative consequences of aging is considered healthy aging. And the gut microbiome has been intensively studied in healthy aging individuals versus unhealthy aging, with clear differences in the comparative analysis of the microbiome profiles associated with these aging conditions. So, this study published by a group in Cork in APC, led by Professor Paul O'Toole, describes the microbiome changes with respect to aging in general and in unhealthy aging. So they grouped the bacteria into 3 groups, group 1, 2, and 3, and demonstrated that the group 1 bacteria were decreased as a result of aging, while groups 2 and 3 actually increased in aging in general. But in unhealthy aging, it was noticed that members of groups 1 and 3 decreased in healthy aging, whereas the group 2 bacteria, which were more pathobionts, were enriched during the aging process. And so they defined the different microbes and functionalities associated with healthy aging versus unhealthy aging. They also looked at adjusting for age. They identified that when adjusting for age led to improved identification of gut microbiome alterations in multiple diseases. So this is a meta-analysis of publicly available data of human cohorts in comparing the genomes of studies conducted in different countries, different continents, as a result of age from relatively young, 25. The cohort started at 25 up to 89, I think it was, and the study conditions were either healthy individuals or individuals with disease at different ages. So the diseases were IDB, type 2 diabetes, liver cirrhosis, colorectal cancer, and polyps. And these individuals were segregated into disease and the different ages. And what they found was that the microbiome was different in the diseases at the different ages. So there was an age interaction with microbiome, which was as strong as the disease in these 2,500 case controls in this experimental hub of a metagenome analysis. So this tells us that age is a driving factor, that's a significant driving factor in terms of microbiome, but, and that it impacts on the disease development at the different stages of life. The same group, led by O'Toole, conducted the ElderMet study and published a few years ago, looking at the microbes, the microbiome alterations in the elderly as a result of elderly condition, whether it was a healthy aged individual or an unhealthy aged individual. So the microbiome was analysed in just under 200 elderly patients aged 65 to 80 years of age, and they were then separated based on where in the community the individuals lived. So obviously the healthy community population were the healthy residents living out in the community, capable of independent living, whereas on the other extreme were elderly people in long-stay hospitals, which were in hospital in a situation like that for at least 1 year. And you can clearly see that there was a clear separation of microbiome composition and functionality, whether the individual was a healthy living community dweller or an unfortunate patient that lived for a long time in a long stay situation. And the elderly health was associated with the community dweller, and they described the microbiome of that, whereas the long stay microbiome was more enriched in pathobionts and was associated with a frail condition in the elderly individual. They further looked at a comparison of these microbiomes with the diet of the individuals, and this is the only animation I have in my talk as far as I remember. So this one, on the green dots linked with the lines there, kind of shows the interaction of